If you want to understand why colon cancer is now showing up in people in their 20s and 30s when it used to be rare before age 50, don't start by blaming genetics. Start by looking at what's on the plate every single day and what's missing from it. For years, we were told that stage 4 cancer means the end of the road, that once it spreads, the outcome is almost certain, and that food plays little to no role in changing that reality. But after nearly 15 years working in emergency medicine and closely observing oncology patients, I've seen something that challenges that belief at its core. I've seen advanced stage 4 colon cancer return to stage 0 by activating the patient's own immune system. I've seen tumors shrink when their blood supply was cut off. And I've learned that the food you eat can either feed a tumor or help starve it. A tumor without blood vessels cannot grow larger than the tip of a ballpoint pen. It stays frozen, limited, contained. But once blood vessels connect to it, that same tumor can grow up to 16,000 times its size in just 14 days. That is the power of access to oxygen and nutrients, and that is exactly where we can intervene. I'm Dr. Becker, an emergency medicine physician. Over the years, I began noticing something deeply troubling. Younger and younger patients were walking into the hospital with colon cancer patients in their 30s, 20s, even teenagers, people who should have had decades of healthy life ahead of them. When I was in medical school 20 years ago, a case of colon cancer in someone under 50 was rare enough to be presented at grand rounds. Today, it's no longer unusual. The incidence in people under 50 has risen by about 50% over the past 3 decades. That doesn't happen because of genetics. Our genes don't change that quickly. Our environment does. Only about 10 to 15% of cancers are primarily driven by inherited mutations. The majority are influenced by environmental factors interacting with our biology. And the most powerful environmental factor? Diet and lifestyle. We are eating more processed food than at any point in human history. Processed meats like hot dogs, deli meats, bacon, and sausage are classified by the World Health Organization as Group 1 carcinogens, the same category as cigarettes. That classification is not subtle. It means there is strong evidence they cause cancer. For every 50 grams of processed meat consumed daily, about 2 slices of deli meat, the risk of colorectal cancer increases by 18%. Think about what happens when processed meat moves through your digestive tract. It sits in the colon for hours before elimination. During that time, compounds such as nitrosamines, heterocyclic amines, and heme iron create inflammation in the lining of the colon. Chronic inflammation is the foundation for cancer. Cells constantly repair damage, but when the damage becomes overwhelming, mutations accumulate. Over time, those mutations can lead to malignancy. But food is only part of the story. A sedentary lifestyle plays a major role. When you sit for most of the day, metabolism slows, inflammation rises, gut bacteria shift in unhealthy ways, and immune function weakens. Even 30 minutes of walking daily improves gut motility, reduces inflammatory markers, and strengthens immune surveillance. Smoking and vaping are also major contributors. Many people assume vaping is safer than cigarettes. That assumption is wrong. The chemicals in vaping liquids can be highly toxic, and they increase colorectal cancer risk. Alcohol is another key factor. Even moderate drinking increases colon cancer risk through DNA damage and disruption of folate metabolism. Here's what all of these risk factors share. They suppress the immune system, and your immune system is one of your body's most important defense systems against cancer. Imagine your immune system as a highly trained security force patrolling your body at all times. Specialized cells like natural killer cells and T cells are constantly identifying and destroying abnormal cells. Right now, as you're listening to this, microscopic clusters of potentially cancerous cells are forming in your body. This is normal. With roughly 40 trillion cells dividing regularly, copy errors happen. Some of those errors create abnormal cells. But in a healthy system, your immune defenses detect and eliminate them before they ever become dangerous. It's like having cameras and guards protecting a building. Most threats are neutralized before they cause harm. But when your immune system is weakened by poor diet, inactivity, smoking, alcohol, and chronic stress, those abnormal cells can escape detection. Once they evade immune surveillance, they still face one more barrier: blood supply. Without blood vessels, a tumor cannot grow beyond 2 to 3 millimeters in diameter. No oxygen means no expansion. Your body naturally regulates blood vessel growth through a process called angiogenesis. Too few blood vessels and tissues die. Too many and problems arise, like diabetic blindness caused by excessive vessel growth in the eye. The body maintains a careful balance. Cancer cells, however, can hijack that system. They release growth factors that stimulate new blood vessels to grow directly into the tumor. The moment those vessels connect, growth becomes explosive. Laboratory research has measured this precisely. Tumors without blood supply remain harmless indefinitely. But once vascular access is established, growth can increase 16,000-fold within 2 weeks. Understanding this mechanism reveals exactly where intervention is possible. And that leads to one of the most important breakthroughs in modern cancer treatment: immunotherapy. Immunotherapy has transformed the way we think about cancer treatment. Unlike chemotherapy, which directly attacks rapidly dividing cells and often harms healthy tissue in the process, immunotherapy works differently. It strengthens and activates your own immune system so it can recognize and destroy cancer cells more effectively. Instead of poisoning the tumor, it removes the barriers that prevent your immune system from doing its job. Cancer cells are not passive, they are highly adaptive. One of their most dangerous strategies is their ability to hide in plain sight. They produce proteins on their surface that act like an invisibility shield, signaling to immune cells that they are normal and should not be attacked. These proteins are known as checkpoints. They essentially tell your immune system, "Stand down." Checkpoint inhibitor drugs remove that shield. They block those deceptive signals so immune cells can finally see the cancer for what it is. Once exposed, the immune system can launch a targeted attack, and in some cases, the results are extraordinary. Patients with stage 4 colon cancer, meaning the disease has spread to distant organs, have achieved complete remission, stage 4 to stage 0. That outcome was once considered nearly impossible. It's important to be clear. Not every patient responds to immunotherapy. This is not a universal solution. But when it works, it can be life-changing. One of the most commonly used checkpoint inhibitors is pembrolizumab, marketed as Keytruda. Others include nivolumab and ipilimumab. These medications are typically given as intravenous infusions every few weeks, sometimes alone, and sometimes combined with chemotherapy, depending on the specific characteristics of the tumor. Not all colon cancers respond equally. Before starting treatment, oncologists test tumor tissue for certain biomarkers that predict response. One key marker is microsatellite instability, or MSI. Tumors classified as MSI high have a large number of genetic mutations. While that may sound alarming, it actually makes them more visible to the immune system once checkpoint inhibitors remove the camouflage. The more mutations a tumor carries, the easier it is for activated immune cells to recognize it as abnormal. Another critical marker is PD-L1 expression. PD-L1 is one of the checkpoint proteins cancer cells use to suppress immune attack. Tumors with high levels of PD-L1 are often more responsive to drugs that block this pathway. If you or someone you care about is diagnosed with colon cancer, it is essential to ask whether the tumor has been tested for MSI status and PD-L1 expression. These tests can determine eligibility for immunotherapy, and that information could significantly influence outcomes. Now, here's where the conversation becomes even more interesting. Even among patients who qualify for immunotherapy, not everyone responds the same way. Researchers wanted to understand why. In 2017, a groundbreaking study conducted at the Gustave Roussy Cancer Center in France examined 200 cancer patients receiving immunotherapy. Some experienced dramatic tumor shrinkage, others saw no improvement at all. The investigators analyzed everything: genetics, age, cancer type, prior treatments. One factor stood out. The only consistent difference between responders and non-responders was the composition of their gut bacteria. Patients who responded well had high levels of a specific bacterium called Akkermansia mucinifila. Those who lacked this bacterium were far less likely to benefit from treatment. This discovery, published in the journal Science, reshaped our understanding of cancer therapy. Akkermansia mucinifila lives in the mucus layer lining your intestinal wall. It plays a protective role, It strengthens the gut barrier, reduces inflammation, and most importantly, enhances immune system function. It essentially primes immune cells so they respond more effectively to cancer once activated by immunotherapy. The research went even further. Scientists took Akkermansia from patients who responded to treatment and transplanted it into laboratory animals that were not responding. Once the animal's gut microbiome changed, they began responding to immunotherapy. The bacteria altered the outcome. This finding carries enormous practical implications. If you are undergoing immunotherapy, the health of your gut microbiome could influence whether the treatment succeeds or fails. And if your goal is prevention, cultivating beneficial bacteria like Akkermansia strengthens immune surveillance long before cancer develops. But there is a challenge. Akkermansia is fragile. A single course of broad-spectrum antibiotics can dramatically reduce its levels. Recovery can take weeks or even months. That means lifestyle choices directly influence whether this protective bacterium thrives or disappears. So, how do you encourage its growth naturally? You feed it the right compounds. Akkermansia thrives on substances called elagitanins, found in foods like pomegranates, cranberries, and walnuts. Studies show that drinking about 8 ounces of pure pomegranate juice daily can increase Akkermansia levels sixfold within 3 weeks. That is a measurable biological shift from a simple dietary habit. Oats and oatmeal also support its growth. So do dried chili peppers, including the red flakes often sprinkled on pizza. Even traditional Chinese black vinegar appears to stimulate beneficial bacterial populations. Pause for a moment and consider what this means. The effectiveness of cutting-edge cancer treatment therapies costing thousands of dollars per infusion can be influenced by the microbial ecosystem inside your gut. That ecosystem is shaped daily by the food you choose. And while immunotherapy represents a major breakthrough, it is only one part of the equation. There is another pathway that directly targets tumors themselves by preventing them from ever gaining the blood supply they need to expand. That next pathway focuses on something cancer cannot survive without: blood supply. Earlier, we talked about angiogenesis, the process of forming new blood vessels. Under normal conditions, your body regulates this process carefully. You need new vessels to heal wounds and maintain healthy tissue, but excessive growth can cause serious problems. Cancer cells exploit this balance. They release signals that stimulate new vessels to grow directly into the tumor, delivering oxygen and nutrients that allow rapid expansion. Pharmaceutical companies have spent decades developing drugs that block this process. One well-known example is bevacizumab, marketed as Avastin. It works by targeting a protein called VEGF, vascular endothelial growth factor, which tumors use to attract blood vessels. By blocking VEGF, these drugs can slow tumor growth and are now standard treatments for colorectal and several other cancers. Here is what surprised many researchers. When scientists began testing common foods using the same laboratory methods applied to pharmaceutical drug development, they found that certain foods demonstrated powerful anti-angiogenic activity. In other words, they helped block abnormal blood vessel growth. Green tea extract, for example, showed such strong inhibitory effects in lab studies that if researchers had discovered it as a synthetic compound rather than a plant-derived one, it would likely have been fast-tracked into drug trials. The active compounds responsible are catechins, particularly EGCG. These molecules interfere with the signals tumors use to attract blood vessels. Tomatoes contain lycopene, a compound with similar properties. Cruciferous vegetables such as broccoli and Brussels sprouts provide isothiocyanates and sulforaphane, which also disrupt angiogenic pathways. Soy foods contain genistein, another powerful inhibitor of tumor blood vessel formation. Extra virgin olive oil supplies hydroxytyrosol and oleocanthal, compounds that reduce inflammation and influence vascular signaling. Omega-3 fatty acids from seafood further support this anti-inflammatory, anti-angiogenic environment. These are not small or theoretical effects. Large population studies consistently show that people who regularly consume these foods have significantly lower cancer rates. In one long-term study following 68,000 women for more than a decade, those who drank green tea at least 3 times per week had a 27% lower risk of colorectal cancer. Daily consumption was associated with even stronger protection. That level of risk reduction from a simple habit is clinically meaningful. When you understand these mechanisms, immune activation, gut microbiome support, and blood vessel inhibition, the question naturally becomes practical. What exactly should you do? What quantities matter? What pattern creates measurable change? Let's begin with the foundation: fiber. This is non-negotiable. Dietary fiber feeds beneficial gut bacteria, reduces inflammation, accelerates intestinal transit time, and minimizes how long potential carcinogens remain in contact with the colon lining. It also directly supports immune function. A landmark study in melanoma patients receiving immunotherapy revealed that for every additional 5 to 6 grams of dietary fiber consumed daily, mortality dropped by 30%. 5 to 6 grams is roughly the amount found in one medium pear. Current guidelines recommend at least 25 to 30 grams of fiber daily for women and 30 to 38 grams for men. Yet most people consume less than 15 grams per day. That gap matters. The most efficient sources of fiber are beans and legumes. Black beans, pinto beans, chickpeas, lentils, and white beans each provide around 15 grams of fiber per cooked cup. Eating 1 cup daily covers nearly half of your recommended intake. If that feels overwhelming, start gradually. Begin with half a cup 3 times per week and increase over time to avoid digestive discomfort. Whole grains also contribute significantly. Oatmeal, brown rice, quinoa, barley, and whole wheat products all provide fiber and additional plant compounds that nourish gut bacteria. 1 cup of cooked oatmeal supplies about 4 grams of fiber and supports Akkermansia growth. Fruits and vegetables add fiber as well, though larger volumes are required to reach daily targets. Berries, apples, and pears are strong choices. Cruciferous vegetables provide fiber along with potent cancer-fighting compounds. The key is consistency, not occasional effort. The second core component is polyphenols. These plant compounds reduce inflammation, feed beneficial bacteria, and directly interfere with cancer-related pathways. Deeply colored fruits are particularly rich in polyphenols. Blueberries, strawberries, raspberries, and blackberries contain anthocyanins that support vascular health and immune function. Aim for 1 to 2 cups of mixed berries daily. Pomegranate is especially valuable because of its elagitanins, which encourage Akkermansia growth. 4 to 8 ounces of pure pomegranate juice or half of a fresh fruit daily is a practical target. Green tea deserves special attention. 3 cups per day, brewed properly, provide meaningful levels of catechins. Water temperature matters. Around 175 degrees Fahrenheit is ideal. Steep for 3 to 5 minutes. Avoid adding dairy milk, as proteins can bind to the catechins and reduce absorption. A squeeze of lemon may enhance bioavailability. Coffee also plays a role. Regular coffee consumption has been associated with lower colorectal cancer risk. 2 to 3 cups daily provide chlorogenic acid and other beneficial compounds. Extra virgin olive oil is another cornerstone. 2 to 4 tablespoons daily supply anti-inflammatory polyphenols that influence both immune and vascular pathways. Use it generously on salads, vegetables, or whole grains. Dark chocolate, at least 70% cacao, in modest amounts, about 2 small squares, daily adds flavonoids that support vascular health. The third essential component involves omega-3 fatty acids. These fats reduce inflammation and influence angiogenesis. Fatty fish like salmon are well-known sources, but even white fish such as cod and halibut, as well as shellfish like shrimp and clams, contain meaningful amounts. Aim for 3 servings of seafood weekly, about 4 ounces cooked per serving. For those following plant-based diets, ground flaxseed, chia seeds, and walnuts provide alpha-linolenic acid. An algae-based omega-3 supplement can offer direct EPA and DHA if needed. These strategies create a biological environment that discourages tumor growth. But there is one more category of foods that deserve special attention because their potency often surprises even healthcare professionals. That final category is cruciferous vegetables, and there's one detail here that most people completely miss. Broccoli sprouts are not the same as mature broccoli. In fact, the difference is dramatic. 3 to 4-day-old broccoli sprouts contain up to 100 times more sulforaphane than fully grown broccoli. Sulforaphane is a powerful compound that activates your body's natural detoxification systems and influences genes involved in cancer protection. It supports immune function, reduces inflammation, and interferes with tumor growth pathways. You don't need large amounts. A small handful, about a quarter cup of broccoli sprouts 3 to 4 times per week, provides a concentrated dose. You can find them in many grocery stores, or you can grow them yourself in a simple jar within 4 days. If sprouts aren't available, regular broccoli still offers benefit. Focus on the stems, which contain more sulforaphane than the florets. Light steaming for 3 to 4 minutes preserves the compound and makes it more bioavailable. Boiling, on the other hand, destroys much of its activity. Soy foods are another misunderstood yet highly protective group. There has been persistent misinformation suggesting soy is harmful because it contains phytoestrogens. The reality is different. The phytoestrogens in soy, particularly genistein, tend to block estrogen receptors rather than overstimulate them. In many cases, they exert protective effects against hormone-related cancers. Beyond hormonal influence, genistein is one of the strongest natural inhibitors of tumor blood vessel growth ever studied. In experimental models, it has reduced angiogenesis by more than 70%. Population data consistently show lower cancer rates in communities that consume traditional soy foods regularly. The key is choosing whole food forms such as tofu, tempeh, edamame, and unsweetened soy milk. Aim for 2 to 3 servings per week. Avoid highly processed soy protein isolates, often found in packaged products. Now, let's move from what to eat to how to eat. Timing matters more than most people realize. Your immune system and gut bacteria are particularly active at night while you sleep. Providing fiber and polyphenols throughout the day ensures those nutrients are available when cellular repair and immune surveillance peak. Avoid heavy meals within 3 hours of bedtime, as active digestion can interfere with these restorative processes. Consistency is far more powerful than occasional intensity. Eating a large serving of beans once a week won't reshape your microbiome. Eating half a cup nearly every day will. Drinking green tea sporadically provides minimal benefit. Drinking it daily creates sustained activation of protective pathways. Biological systems respond to repeated signals, not isolated events. Preparation methods also influence effectiveness. Tomatoes are a perfect example. Raw tomatoes contain lycopene, but much of it remains trapped within the plant structure. Cooking tomatoes breaks down cell walls and dramatically increases lycopene availability. Tomato sauce, paste, and roasted tomatoes are excellent choices. Adding olive oil enhances absorption because lycopene is fat-soluble. Cruciferous vegetables should be lightly steamed or eaten raw. Overcooking destroys sulforaphane. For green tea, water that is too hot can damage catechins while water that is too cool fails to extract them properly. About 175 degrees Fahrenheit, steeped for 3 to 5 minutes, strikes the right balance. Variety creates synergy. Different berries contain different anthocyanins. Rotating between blueberries, strawberries, raspberries, and blackberries ensures a broader spectrum of protective compounds. The same applies to legumes. Black beans, lentils, chickpeas, and white beans each provide distinct fibers that nourish different bacterial species. Microbiome diversity predicts health outcomes. The wider the variety in your diet, the more resilient your internal ecosystem becomes. If you are currently undergoing cancer treatment, these dietary strategies are designed to support, not replace medical care. Communicate openly with your oncologist. Certain foods can interact with specific chemotherapy agents, though many interactions are beneficial. Coordination ensures safety. If your goal is prevention, these foods should become your baseline. You do not need perfection. You need steady commitment to high-impact choices. Beans or legumes daily. Berries daily. Green tea or coffee daily. Cruciferous vegetables at least 4 times per week. Seafood 3 times per week. Extra virgin olive oil every day. Soy foods regularly. That pattern builds protection. One of the biggest mistakes people make is attempting to transform their entire lifestyle overnight. Radical change rarely lasts. Sustainable improvement builds gradually. Start with one habit. Add a cup of beans to lunch or dinner and maintain it for 2 weeks. Then add a cup of berries at breakfast. After that, incorporate green tea. Step by step, the routine becomes normal. Another common misconception is that supplements can substitute for whole foods. Fiber powders, omega-3 capsules, and polyphenol extracts have specific uses, especially when access to whole foods is limited. But supplements isolate single compounds. Whole foods contain hundreds of interacting molecules working together. A cup of blueberries offers fiber, vitamin C, vitamin K, manganese, anthocyanins, and numerous other phytonutrients. Their combined action cannot be fully replicated in pill form. There is one more point that cannot be ignored. If you notice blood in your stool, persistent changes in bowel habits, unexplained weight loss, or ongoing abdominal discomfort, seek medical evaluation immediately. Early detection saves lives. Screening guidelines now recommend colonoscopy beginning at age 45 for average risk individuals and earlier for those with additional risk factors. Symptoms should never be dismissed. Colon cancer is highly treatable when identified early. It becomes far more dangerous when diagnosed late. Prevention and early action work together.